OK, bonjour tout le monde, on va commencer. Bienvenue à cette conférence de l'IBIS. Aujourd'hui, on a la chance de recevoir professeur Omer Gokcumen de l'Université de Buffalo. Dr Gokcumen est professeur agrégé à cette université depuis quelques années. Il a débuté en 2013 sa carrière indépendante. Et puis avant d'avoir son propre labo, il a fait un stage postdoctoral à Harvard où il a étudié la variation structurelle dans les génomes de primates. Et ce travail-là a suivi un doctorat qu'il a fait précédemment en génétique des populations humaines à l'Université de Pennsylvanie, où il a étudié différents sujets reliés à la variation génétique dans le génome humain. Donc, depuis quelques années, il s'intéresse toujours à l'évolution du génome humain et du génome des primates. Il a fait des travaux super intéressants sur la variation structurelle de certaines familles de gènes, dont, entre autres, les gènes qui codent pour l'aminase, qui sont à la base de notre système digestif. Il a fait des découvertes intéressantes sur l'évolution répétée du nombre de copies des gènes qui codent pour l'aminase dans différentes espèces de mammifères. Ces exemples-là, c'est des exemples qu'on retrouve maintenant dans les textbooks et dans les cours en génétique puis en biologie de la cellule, tellement ils sont élégants, puis ils nous parlent concernant l'évolution, l'importance de la variation structurelle et de la duplication des gènes. Donc aujourd'hui, il va nous parler de comment l'alimentation au cours de l'histoire humaine a contribué à modifier l'architecture dans notre génome. Et donc, je suis très content qu'il soit avec nous aujourd'hui. Omer, the floor is yours.
Okay, thank you very much. I did not realize that a lot of the names that I use in my work and thinking is actually there. That is like I never make the connection between the actual names in the papers. And it's great to be here and share some of our research with you. I'm going to talk about first about our general thinking about our problems. And the really big and in a way silly question that we are asking is what makes us human? That's the driving question for my laboratory. And it's a silly question because it's such a large question. It's very difficult to come up with testable hypotheses about such a large question. So we need to narrow it down. And it's also a subjective question in the sense that we're interested in it because we are humans. If you're a biologist, for example, and if you're interested in genome evolution, probably it's not a great model organism. You want to work with plants, you want to work with sticklebacks, you want to work with seaweed flies. So but it is actually, because it's subjective and because of its us, genomics, in my opinion, provides a really nice objective way of thinking about humans. At least like it will give us some empirical handle as to human variation and human biology. For example, independent of all the narratives about the role of gender, males, females, etc., we know from a biological point of view that we have a sex chromosome, we have sex chromosomes, and these sex chromosomes have different properties and different biological meanings. And similarly, we can actually work the genomes in a cheap and very efficient manner nowadays. And one of the things that happened in the human evolutionary research, we are always late to the party. Almost always people figure out certain genomic elements in other organisms, and suddenly there was a big flashy paper showing that, oh, look, like there's structural variations that, not SNPs, but other type of variations are also important. And that actually happened about a decade ago in the human genomics, and there was a huge explosion of data sets. But it turns out that there's a lot retrospectively, of course, but now like we know that there's a lot of variation that fit into this general term, structural variation, which includes deletions, duplications, inversions, etc. And we actually published a couple of papers on that as well. One of the things that is clear is that the sheer size and amount of structural variation of defining underlying the human and primate genetic variation overall. So in fact, SNPs are almost negligible when it comes to amount of variation that we are looking at when we consider base pairs affected. So with that general idea that my laboratory's focus is a little bit narrowed down to the argument that structural variation really underlies the phenotypic variation in humans, that's where the gold is, if you will. So that has been our focus for the last 10 years or so. And one of the great things now we can do is that my laboratory generates a lot of data sets, long read data sets, short read data sets, transcriptome, proteome, regulatory ATAC-seq data sets. We also integrate all the data sets available. And that's a wealth of data, but it creates what I call a library of Babel problem, which is this idea that Borges put forward. I really like that metaphor, that there's a library and all the information of the world is available in the books in this library. However, there are no index cards. So people don't know where to look. They don't know which books actually contain the information. So they're essentially lost. And in practice, what that means is that they know nothing. And in a way, in this wealth of data, without the evolutionary thinking, that it will be very hard to navigate this gigantic amount of omics data. So my laboratory overall uses these very high throughput data sets, combine them with population genetics and evolution genetics methods and questions, and then come up with specific hypotheses to test, which we generally test within the context of either mouse models or cell models or some other, functional approaches. So now I'm going to move on to my actual talk and to lay the groundwork. One of the things that we found, looking at all the human genetic variation and our analysis, shows that metabolism and the genetic variations that relate to metabolism seems to be a major driving force in the human evolution. And of course, when you talk about metabolism, it immediately means that you're talking about diets. And diets is such an interesting thing in humans because unlike some other organisms, in very recent human history, our diet has changed multiple times and in an extremely drastic manner. That while we were eating protein-heavy diets, for example, at some point, we switch, some populations switch completely to carbohydrate-based diets. Our ancestors in the African rainforest, chimp human ancestors, were probably eating forest-based, fruits-based food. We move into the savanna, and then we become likely hunter-gatherers, and our food diet change. In the modern era, our diet change again multiple times, first with agriculture and after with the Industrial Revolution and mass production of the foodstuff. And the question becomes, these kind of cultural shifts, do they affect the metabolism, our perception, our energy consumption? What happened? And is there any signatures of those in the genome? And the short answer to that is yes. So for example, we just finalized a paper with Amanda in Calgary, and we looked at the differences in the repertoires of olfactory receptors and taste receptors in Filipino and Ugandan hunter-gatherers and farmers. And we show that there's huge amounts of individual variability in the active and non-active olfactory receptors, either due to structural variations or loss of function mutations, so that in any given individual, including us, would have a different repertoire, what they can perceive and what they cannot perceive with regards to olfaction and taste. And some of them, we actually published a paper with Kara and show, for example, that there seems to be diet-related selection acting on at least some of these olfactory receptors. One finding very interesting, and it's a little bit hand-wavy, but I think it fits, that one particular olfactory receptors that is sensitive to boar testosterone actually seems to be selected against in populations who actually domesticated pigs very early on. So you actually see huge population differentiation in that particular olfactory receptor that is linked to having a negative reaction or negative smell associated with male pigs and male boars. And then, since that perception issue is important and it seems to be changed due to, at least to some extent, due to our dietary change, what we actually end up doing is looking at spit, which seems to be a little bit underappreciated part of our biology, because the spit, the saliva, is the fluid where we actually put something in our mouth, and that seems to be the main place where digestion starts, where perception starts, and where immunity starts. So we think that it's an important fluid to understand what's going on. And one of the things that we did, this is a paper that we published last year, showing at the transcriptome and proteomic levels, you have this finely tuned machine, if you will, coming from 3 different salivary glands, pumping up all these proteins into the oral cavity, creating this like pretty sophisticated array of proteins, including amylase, which I'm going to talk about, but some others. And then we actually continued this work by doing the same thing with mouse, which is actually ongoing. We didn't publish it yet, but one of the most amazing thing is that almost none of these proteins exist in an identical by descent fashion in the mouse saliva. So somehow there's a huge turnover that, mouse has like some other proteins that replace these. So something happened between human evolution and mouse evolution that leads to these differences in the salivary proteome. So that's kind of interesting. So we wanted to understand this process a little bit more. So we started to look at differences between different primates. And what we actually found, that even though it's not as drastic as the difference between mouse and human, we actually see pretty visible differences, especially in the dosage of the salivary proteins between humans and non-human primates, which further the idea that saliva may be evolving pretty fast because the diet is changing very rapidly, especially in the human lineage. And I will talk about amylase, which I basically highlighted here. But I think I wanted to cap this section saying that there's an amazing diversity of salivary proteome across species and between humans as well. And I think thus they actually signify differences and outcomes in immunity, metabolic processes, and maybe as a result of this diet change idea. So Petar Pajic, who is a PhD student of mine, actually led this study. I'm going to very briefly mention this because amylase is a fantastic, really interesting locus. The initial finding was that there's huge number of structural variants in the manner of copy number variants of that particular gene. So the idea is that almost all mammals have a single copy of that gene, and that gene is an important gene, conserved gene, that is expressed in pancreas and is primarily involved in digestion of starch. And the story goes, there was a paper published about a decade ago showing that there's an expansion of the amylase genes, and the copy number increases, but the actual sequence was conserved. So it's not neofunctionalization, but rather the same gene being preserved, and that it leads to a gain of expression in the saliva and further copies actually increase the dosage of that gene's activity in the saliva, and people actually link that with agriculture, a shift to agriculture. However, what we actually realized is that people actually publish a paper on dogs. People actually show that in the saliva of the mouse, there's some amylase copies, the amylase activity. And in rhesus macaques, for example, there's actually more amylase in rhesus macaque saliva than in humans. So that was very curious. So Petar ended up collecting samples from 68 species, hundreds of samples. And what we found is that instead of this very simple picture, we have this massive amount of variation between species. And there's at least 4 species, dogs, pigs, rats, and mice that actually have human-like copy numbers of the amylase. And all of these species have abundant salivary expression. And of course, you can immediately see the connection here that there's a commensalism issue, that there may be a recent, actually in dogs, it was already shown pretty clearly that this actually happened in the last 20,000 years or so, when these animals become commensal and then start eating human foods, essentially. Not only that our culture it seems that is changing our genomes, domesticating our genomes subconsciously, unconsciously, but it's also changing the genomes of other species. I'm not going to get into that, but I'm sure that, for example, Christian will be interested in some of the insight that we gained about how on earth these guys are recurrently gained over and over again from a single copy, and how on earth that that these species independently gaining the saliva expression. So we have some insight into that, which involves retrotransposition that is accompanying these gene duplications. We also have this neat analysis done where we actually show that the likely digestion is not the main driving point. It seems that perception at the molecular level and at this psychological level seems to be the driving force. You want to have saliva amylase in your saliva so that you can taste starch, both at the molecular level, so that you can tell your brain that you're eating something sugary, and also psychologically that you actually want to eat those bread. So with that, I will basically cap this part. And the idea here is that culture seems to be affecting genomes with implications to our immunity, metabolism, and perception. I did not talk about immunity, but I will in a minute. Any questions so far before I get to the main bulk of my talk? We're good.
Omer, maybe one quick question.
Yep.
Yeah, I mean that's a fascinating pattern with the increase in copy numbers, dogs, pigs, humans. What about the homology?
So the homology is very, very high, but we showed using actually retrotransposon tags that they are independent events. They are not. So as far as we can tell, the ancestral mammal has a single copy, and you have at least 4, probably 5 or 6 bursts of duplications that happen independently. Of course, the question becomes, what is special about the amylase locus, and why the mutation rate is so high, which we are kind of working on right now. Not kind of, we are working on it with longer reads to understand exactly when those events happened, etc.
Okay, thank you.
Okay, so now I'm going to move on. I basically make the case about diet and these structural variations. So one thing that we wanted to do, I'm going to talk about this particular paper, which is recently published. We really wanted to understand if there are really old variations that may be linked to these dietary changes, these metabolic changes. And to do this, we did a genome-wide analysis of deletions. And we were working with deletions because it's not impossible, but it's very difficult to work with duplications and inversions in the human genomes with this current technology, because the genotyping is not very well. But with deletions, we can actually genotype them pretty well and find the linkage disequilibrium islands, the haplotypes, so that, for example, we can measure the date of them. So here I'm showing you on the x-axis from the age of the haplotypes carrying deletions. And on the y-axis, I am showing you different categories of deletions. We actually use Neanderthal and Denisovan genomes, and we genotyped human polymorphic deletions in these ancient genomes. And we then categorize them. If they're shared, why are they shared? Are they recurrently evolving in Neanderthals and humans independently? Are they intergressed, meaning that are they admixed from Neanderthals into humans recently? Or that they're actually shared because they share a common ancestor way back when, and the probably Homo erectus population actually have variable deletions, and these variable deletions remain variable since today. And based on these categorizations, we actually identify hundreds of really, really old variations that still linger in our genomes. And then we actually modeled that this is actually, there's an excess. Like it's, there's an excess of allele sharing of these ancient deletions. And the idea or the hypothesis here is a little bit controversial, but the idea is that maybe balancing selection of sorts, maintaining at least some of these deletions. So then the next thing, then we basically search for metabolically or developmentally or dietary related genes that may be affected by the deletions. And one thing pop up, and that is the growth hormone receptor, a deletion in the growth hormone receptor. And the reason why this was weird is that growth hormone receptor is exactly what it sounds like. It is the receptor, the main receptor for the growth hormone, and thus it's a very important gene. And when it comes to important genes, you don't expect them to carry exonic deletions, polymorphic and common. So as you can see that, it creates a dimer. It receives the growth hormone, which is done by the blue. This part is the growth hormone receptor, and it actually activates very important pathways, which leads to transcription of target genes and growth and metabolism. And if you have a knockout of this gene, you actually end up certain type of dwarfism and developmental delay in humans. So, and as expected, the coding sequence of the amino acid sequence of that particular protein is very conserved among mammals, like anteaters and humans pretty much have the same amino acid sequence, but somehow half of our chromosomes as humans is missing these 20 plus amino acids. So some of us actually have shorter version of growth hormone receptor, which is very odd. Like you wouldn't expect such a conserved gene, such an important gene, to have such a drastic polymorphism that is very common. So we wanted to understand what's going on and how did this particular deletion evolve. And of course, like we want to understand if there's any kind of adaptive meaning to this. So first thing we looked at is that this deletion is very common. It's actually the major allele in Africa, and it is a little bit less in Eurasia, but it's still like, it's still above like, 10%, 15% allele frequency, pretty much in all human populations that we looked at. As I mentioned that some of us actually carry this homozygously, some of us carry heterozygously. There's no deviation in Harder-Weinberg. And all of the Neanderthals and Denisovans that we looked at actually have that deletion. So that it seems that it is fixed in Neanderthal and Denisovan lineage, but it's polymorphic in humans. So then the idea is like, it kind of looks like, it evolved maybe 1.5, we actually dated this to 1.5 million years ago, the emergence of that deletion in the human lineage. And it actually increased in allele frequency to the extent that it is actually fixed in some of our cousins. But somehow it remains polymorphic in humans for a long time. So what we actually did is we worked with Leo, and he really has a brilliant method called relate. And this particular method shows that can actually predict allele frequency trajectories across time. And one thing that his method found, and we actually did it in an alternative Bayesian way as well, but I think this summarizes well. It seems that it was very high allele frequency for a long time in the human populations, but then it decreased very recently, drastically. In fact, it was so drastic that we couldn't explain it with this drop in East Asia and West Eurasia. We couldn't actually explain it by neutral processes, that this has to be selection against the deletion. So it was kind of a curious story that it doesn't fit to normal classical sweeps because it's not fixed in the humans. It doesn't fit with balancing selection like a heterozygous advantage because it is actually not steady across time. So it is more complicated than that. So what it looks like is this, that you have this very old mutation increase in allele frequency and got fixed in archaics and remain polymorphic and very high allele frequency in the human lineage, only to be go down in allele frequency in the very recent years, creating the current allele frequency landscape. So then if there's a fitness advantage, then what is it? So that's like the next thing we did. And the first thing we did is we did a meta-analysis of genome-wide association studies available. So there's actually a huge amount of data now. So what we can do is that take that haplotype, even though this particular deletion is not genotyped, we can actually take tag SNPs, that is so that we can essentially impute the deletion and then look at the traits that it is associated with. And it turns out that it's small birth size, small placenta size, time to sexual maturation, bone ossification, and longevity were the traits that come up significant. And that makes us extremely confused because I'm not an ecologist. I guess, like, you know, it turns out that if you're an ecologist, that will actually be are very meaningful to you, because it took us about 6 months to figure out what's going on. And this particular paper really was insightful because the idea is that they have this, the argument is that under resource limitations, compensation for growth, like, you know, early small start and compensatory growth seems to be a strategy in many organisms to actually deal with starvation and nutritional deficits. And they have a very, very helpful list where they actually see what kind of traits that are affected within that Evo-Devo kind of setup. And these are the traits. All of our traits were in that list, that there seems to be something something to really think about. And starvation resistance is something that we didn't think, but it seems that it fits very well. So what we did next then is we actually looked at this particular disease. We were lucky enough to have access to this Malawi cohort, which is they're suffering from severe malnutrition. And it turns out, I did not know that actually, that there's a particular metabolic syndrome following severe malnutrition. Not everybody was affected, but if they're affected, that actually leads to some sort of swelling of the belly, like, loss of fluid intake and a lot of problems. And not everybody, there's a genetic background to how susceptible to you are. And we actually managed to genotype that deletion in this cohort and show that indeed the deletion is actually significantly protect you against that particular syndrome, that you're better off during starvation if you have the deletion. So that was actually a pretty big finding. We were very excited. And at that time, then we were talking about what to do, etc. And then people really want me to look at the mechanism because it was great, but we have absolutely no idea about the why there's an association, but why this is happening. So what we then did is, okay, like, since this is a very conserved gene, what we could do is that mimic the deletion in a mouse model, because the mouse actually has exactly the same sequence. So we deleted that particular sequence, essentially mimicking the human condition, and we generated all these cohorts, which is normal diet, male, female, wild type, and homozygous deleted mouse. And we did the same kind of cohorts, but this time for color restriction to mimic or to model the starvation situation. And what we found is a pretty remarkable lack of sexual dimorphism in males, between males and females for the deleted mouse, the D3 mouse, under color restriction conditions. So you can actually see that this is the normal diet wild type mouse, and you can see that males are 30 to 40% larger than females as expected, and there's no effect of the deletion under normal diet. So it seems that there's nothing is happening for growth. But under color restriction wild type mice, there's a shrinkage, like they both become smaller, but males become even more smaller. So like there's some shrinkage of sexual dimorphism. But with the deletion mouse, the actual sexual dimorphism completely disappears, because actually this is a significant observation that the males are smaller in this case under color restriction significantly. So that was a really interesting finding, and we basically explain it at the expression level, looking at liver expression. What we actually found first, these are example genes. Again, it's the same idea. This is the normal diet. This is color restriction. We have females and males, wild type D3, wild type D3. And what we are actually showing here is that this particular gene, Cry2, is a very well-known central circadian rhythm gene. And it turns out that only in males and only in normal diet, you actually have an increase of that particular gene. It's very clear. It is as if the males are under color restriction if they're carrying the deletion under normal diet. And what we actually see afterwards is that a second trend that we show is that male bias genes become downregulated under color restriction if they're carrying the deletion. They become female-like, if you will. And the female bias genes become upregulated. So what I'm trying to say here is that under normal diet, there seems to be no such effects, but the under color restriction, the GHRD3 mice, the male mice, expression seems to become female-like. And we have shown that this is actually showing that this trend is true, like these trends are true at a transcriptomic level. It's not just one or two genes. What we are showing here is that the fold enrichment, when we compare color restriction to normal diet, is extremely well correlated with the difference under normal diet between the deleted mouse and the wild type mouse. In other words, if something is upregulated in color restriction, that's also upregulated in mouse with that deletion happening. So the deletion effect is mimicking the effect of color restriction in male mouse. And when we look at the actual mechanism based on these, that normally if everything is normal in wild type mice under normal diet, we have a pulsating growth hormone like this, which is a characteristics of male growth hormone pathway, which leads to growth hormone receptor, which leads to intermittent STAT-JAK activity, male bias expression, and normal sexual dimorphism. But what happens in color restriction is that you have this pulsating GH become like, less pulsating, and to compensate, that you have there's already published circadian rhythm-based compensation mechanism that actually increases cyclical growth hormone secretion in males. And that is why we actually see the similarity between the deletion and the color restriction effects, where the circadian rhythm compensation is trying to compensate the effects of the growth hormone receptor D3 because it creates less pulsating growth hormone signal downstream. So with the female base under color restriction, however, we actually see no differences in these circadian rhythm difference business. But what we actually see that the growth hormone receptor D3, GHRD3 mice seem to act like female. The livers of the male guys under color restriction acts as if they're females. And the explanation here is that the color restriction already spans the compensation, if you will, from the circadian rhythm stuff that's already like trying to compensate the effects of color restriction. What happens is that color restriction plus the GHRD3 ends up leading to loss of this pulsation, so it's dampened, and it leads to persistent stat-JAK activity instead of intermittent stat-JAK activity, which leads to female-like expression, which leads to lack of sexual dimorphism. So that is cool. So like, now that we know that this protection against starvation is probably due to female-like expression in liver and probably other places in the body, which leads to metabolic changes. So which then the development and metabolism of the male mice becomes more female-like, leading to smaller body sizes. So then the question becomes, okay, like, that's great, but why is it, what is the balance? If it's good for starvation, why not everybody actually have the deletion, and some of us actually have the deletion and some of us don't? And we basically thought that maybe there's some sort of sexual selection happening, that, bigger males have like, better fitness because they have more kids, etc. But the clues actually comes from a very recent work that we did with Holly Ingraham lab at UCSF. Joni actually led this effort. It's like a beautiful study where they actually show a particular conditional knockout of this gene called BCL6 to be mimicking the effects of GHRD3, but in a much more drastic fashion in the mice livers. Here I'm showing the GHRD3 effect size as compared to controls under color restriction, and this is the BCL6 conditional knockout mice expression levels in liver, and you can see that the effects are almost identical. It's like highly correlated, and this like GHRD3, it creates female-like liver expression in the male mouse. And why is this important? Because what they're showing is that in males and females, the metabolism is very different when it comes to liver. That fetal liver disease seems to be primarily, it's a sex-specific disease in humans and in mammals likely. And after overeating, you actually, in males, develop this fetal liver, which they can actually rescue, that you don't actually develop it if you have the BCL6 knockout. And this effect is also very similar to what we did with the mouse serum lipids, that we actually show that there is the GHRD3 leads to lower lipid content in serums in male mouse. And it's actually the effects are opposite in females and males. And now why this is important, this actually fits well with the whole starvation business, right? Like, you have more fat accumulation in the liver in males And if you actually interfere with that particular male specific trait, then you actually have less fat accumulation. So you're smaller, like the idea actually fits well. But what is more interesting is that they actually link that to infection, where they show that this particular TAG accumulation, triglyceride accumulation in serum, seems to be affecting females more than males under normal conditions. But in the when you actually have the knockout, this growth hormone pathway interference, then you will actually have a female-like TAG accumulation after E. coli. So the response to infection is becomes more female-like in the male animals. And they show that at the survivability level, and they show that in general, and these are stressed animals because the temperature is modified, but they show that under these stress conditions, males, normal wild type males, deal with E. coli infection much better than females. However, when you actually do this knockout, when you interfere with the growth hormone pathway, you actually have a huge decrease of survivability. This is conditional knockout, and this is non-activated. This is still transgenic mouse, but it's non-activated, and this is activated. So you can see that in females, this particular pathway interference doesn't actually make a difference. So then the idea becomes that, yes, you are actually, by interfering with growth hormone pathway, and this interference can be with growth hormone receptor D3 deletion, or there may be other genetic variations that may also interfere at the genetic level with this pathway by changing the dosage of some of the proteins, etc. But this pathway seems to be balanced. On one side, surviving better when there's less food at the metabolic scale, and on the other side, you have this vulnerability or resistance to infections. I guess then both of these issues, resource availability and the pathogenic pressure, are dependent on time and place. So thus it makes sense that you actually have this oscillating selective coefficient changing the adaptive landscape for both the deletion and probably some other genetic variations affecting this particular pathway. So of course, the one question that we could not answer still is why males? Why is this just a evolutionary, one of those chance events that it happens to affect the males and it worked out fine? Females are already relatively resistant to starvation. We move on. Or is there a particular adaptive reason why males specifically being targeted by this adaptation? I think there needs to be some more studies to answer that particular question. So just to wrap up my talk, we are actually showing the clearest example of a sex-specific and environment-dependent effect of a common polymorphism. I know that this is actually shown in insects and fish and some other mammals, but we don't actually have such a clear example in humans yet. So it was, I think, as far as I know, one of the first ones. And it also exemplifies how human evolution is convoluted by culture, gigantic amount of migrations going into different ecological niches, and the timing, meaning that what the adaptive challenges 50,000 years ago is not the same as the adaptive challenges 10,000 years ago, and those are not the same as today. I think the last point that we can make is that these kind of evolutionary signatures, evolutionary history, has major effects on human health today, because we are carrying these variations in our genomes today with unknown ramifications for obesity research, for fasting. Like there's intermittent fasting, for example, right? It's common. everybody says that, oh, it works great for me, and sometimes it doesn't work. Maybe it is related to this growth hormone receptor pathway related processes. So another thing that this is kind of a pet peeve of mine, but I like that idea that Levantin made this pretty neat observation that majority of genetic variation in humans is within populations and not between populations. And I think that's a good reminder that there's no biological meaning in races. That doesn't mean that races are not real. I'm saying that biologically it doesn't mean anything when it comes to humans. And not only that, this variation that we share between different populations, a lot of them are actually really, really old, that we actually have those variations before we were humans in a way, that, while we were Homo erectus. So this is part of our shared legacy, this diversity, this variation, which I think we should embrace and think about in this odd times. And the last thing to close out, that I would just want to say that structural variation is really cool. There's a lot of the structural variation we still do not know, especially the inversions, short tandem repeats, exonic repeats. Like, we really don't know what's going on in the human genomes. And with long reads and better assemblies, I think we will actually at the edge of an explosion of understanding of missing heritability and some of the traits that may be adaptive, and we didn't even know that because we just couldn't see, and now we can. So I'm very excited about moving forward in the coming years. With that, I'm happy to discuss any of the topics, and I'm always recruiting. So if anybody is interested in living in Buffalo, it's good. Cold is not an issue anymore, and we have great food. All right, guys, thank you very much.
Thanks a lot, Omer, for your great talk. That's a fascinating story.